Good afternoon, everyone. My name is Natthawut Pratumwan. I am master degree student from Computational and System Medicine Research Group of Faculty of Medicine, Khon University. Today, I would like to share about my research project about the improving efficiencies of cisplatin through CRISPR technology in cholangiocarcinoma cell line. And this is the overview of my talk today. Begin with, I will introduce about the cisplatin resistance in cholangiocarcinoma, and followed by the CRISPR-Cas9 based chemoresistant gene knockout. And the main point of today is the characterization using the multi-omics approach. And some data of my project is the data from the structural variations analysis. And beginning with the cholangiocarcinoma. Cholangiocarcinoma is an epithelial cell malignancy that have the varying incidence worldwide. And in Thailand, especially in the northeastern regions of Thailand, has the highest incidence or highest number of cases of CCA. And the risk factor for CCA in northeastern regions of Thailand is the river Phuk, Opisthochis viverrini. And nowadays, we have many treatments for CCA patients, including the surgical resections, which is commonly used for CCA patients. And another treatments, including the chemotherapy, radiotherapy, immunotherapy, and the targeted therapy. And for the chemotherapy, the cisplatin as the first-line chemotherapeutic drugs is also used for the post-operations or post-resectable CCA patients, because of some resectable patients have a high frequencies of relapse and metastasis after surgery. But the number of CCA patients who benefit from adjuvant therapy or the cisplatin is low because of the resistance to cisplatin in CCA. And we have the 5 possible mechanisms that CCA can resist to the cisplatin to escape from the effect of cisplatin, including the cellular drug accumulation, the detoxification system, DNA repairing system, apoptosis, and the autophagy. And today I will give one example of the cisplatin resistant mechanism is the cellular drug accumulations. And this figure shows the journey of the cisplatin begin with the entering into the cell until it exporting out from the cells. And firstly, the cisplatin will enter to the cell through the CTR1, that is the drug importing protein, and then it will exported to the cisplatin exporting protein ATP7A and ATP7B that located on the Golgi body, and then it will be exported out from the cell that causes the decreasing of the cisplatin. That is the mechanism of the cisplatin resistance in CCA. And I will give some example of some research that use the CRISPR-Cas9 gene editing technique to knock out the drug exporting protein in the cancer cells. And this research, they use the CRISPR-Cas9 to knock out the ABC transporter proteins, which is the protein to export the drug from the cell that can be the key protein for the drug resistance. And the CRISPR-Cas9 has the potential to knock out this protein and then can improve the drug sensitivity after knock out this protein. And next, I would like to talk about the multi-omic approach. So the multi-omics or omics data is the methodology from the combinations of many omics data to discover the complexity of biological systems since from the beginning with the genes level until the metabolisms that can indicate the phenotype of the organisms. And how are omics or multi-omics important for CCA studies or other cancer studies? Because of cancers or CCA have usually have the DNA mutations or the dysregulations of the epigenetic regulations, and also the protein-protein interaction. Some protein can interact that can help to increase the survival rate of the cancer or CCA, and also with the metabolism of the cancer that can be the one of major factor for the drug resistance, because it can detox to decrease the effects of the drug when the drug stay within the cells. And from these reasons, this is the reason why omics important for the cancer study, because if we can discover about these informations of the cancer, we can use this information to understand the molecular mechanism of cancer, and also can decide about the new treatment to target the target protein or target gene, or even the metabolite within the cancer cells. And then and from these reasons, many omics data is very important, because if we integrate many omics data together, we can answer about the questions of the cancer or each cancer in every level. And the interesting thing is the integration of many omics can answer about the drug response of the cancer cells, or to understand the molecular mechanism of the cancer cell, or even in the case of CRISPR-Cas9 gene editing technique, if we knock out some protein that involved with the metabolisms, it will show the different phenotype when compared to the wild type cancer cells. And this is my project journey, and it have about like 4 bus stop. For the first bus stop is the characterizations of wild type CCA cell line, and then followed by the evaluations of cisplatin resistance, and then I will use the CRISPR-Cas to knock out the chemo-resistant genes, and then I will characterize the cells with the gene knockout compared with the wild type of CCA cell line. And for the first, and now I am in the process of the first one is the characterizations of CCA cell lines using the genomics, epigenomics, proteomics, and metabolomics studies. And for today, I would like to talk about the analysis of structural variations. And for this study, I used 4 cell line models, including 3 CCA cell lines, KQ21TA, KQ21TC, KQ452, compared to the human cholangiocarcinom cell line MMNK1. And then the genomic DNA extracted from those 4 cell lines was used for the whole genome sequencing using the 3rd generation sequencing Oxford nanopore. And then the nanopore data was used for the variant calling using the structural variation calling algorithm, and then visualize it to see about the variation within the 4 cell line. And this one is the Venn arrow plot for the variant within the 4 cell line. For the left plot show the number of variant position in each cell line, and for in the middle number is the 87 is the number of the position variant that 4 cell lines share together. And when we compare 3 CCA cell line to the cholangiocarcinom cell line MMNK1, it have about more than 60 position variant similar to the MMK1. And for the right figure or the right plot show the SNP position within 4 cell line, and about 34 SNP have the similar position of SNP together of these 4 cell line. And this figure show about the type of the structural variation in the KKU213A cell line, and the most variant is are deletion and insertion. So the size of the variant for the deletion and insertion, it will stay within the range about 50 to 100 sites, and for the duplication and inversion, it will within the about more than 5,000 sites. And for the other three cell lines show the similar data or similar result, that the most variant is the either deletion and insertion, similar to the KKU220A, like this. And then these two plots show the intersections of indel or insertion divisions of four cell line. And for the heat map show the intersection of indel between 3 CCA cell line with the cholangiocarcinom cell line, and it have the different profile of MMK1 compared to these 3 cell line. But it has interesting point that when we see in the different the intersection between KKU220C and KKU220A, it show the highest of the indel intersection value. And if we see on the upset plot, when we see in the intersection size between KKU220C and A, it has the highest size of the intersections of the indel intersection. And for the last one is the relationship between indel intersection of these four cell line. And to summarize the data, it shows that most structural variations of these 4 cell line are insertions and deletions, and the structural variance between 3 CCA cell line compared with the human cholangiocarcinom cell line MMK1 are distinct, and the intersections of KKU220A and KKU220C have the highest value because they come from the same type of CCA, is the intrahepatic CCA. And from this result, it is the overview of the variance of these 4 cell line, and I have to study more about the variant within the each gene in this 4 cell line, and also discover about the more omics data to integrate them, and it will show more clearly about the informations of these 4 cell line. And I have many thanks for my advisors, Assistant Professor Juthalop Pechalaburunin and Dr. Paramet Ganwit, and Cholangiocarcinoma Research Institute for my research grant, and all members from Computational and System Medicine research group. And that's all of my talk today, and thank you for your kind attention.